Hi students, how are you? I hope each one among you are doing great and well. Welcome to Supnit's classes. This is Rahmat Latif, your biology educator. Today we are going to learn post-fertilization events. Okay, what are post-fertilization events? I'll try to explain. What are post Actually, we have three events. Mainly, we have three events. Okay, be that in animals, humans, or be that in plants. We have three events mainly. First event is pre-fertilization event. Fertilization events. Another event is fertilization events. And finally, we have post-fertilization events. Okay, post-fertilization events. Pre-fertilization events are those events which took place before fertilization, okay? Then we have post-fertilization events. Post-fertilization events are those events which took place after fertilization. Pre-fertilization events took place before fertilization and post-fertilization events took place after fertilization, okay? We are done with pre-fertilization events. We saw fertilization events. Now we will saw post-fertilization events, okay? I'll erase it. In this session, we are going to see what are post-fertilization events. Let's see. Let's see what are post-fertilization events. Post-fertilization events. Okay. As I already mentioned, post-fertilization, these are those events which took place after fertilization. Okay. These are those events which took place after fertilization. Okay. Now we will see what kind of post-fertilization events took place in plants. Okay, we will see what will happen after fertilization, what kind of post-fertilization events will took place. Okay, first of all, we will see. We saw that in plants double fertilization occurs, right? We saw double fertilization occurs. In our previous session, we saw double fertilization occurs. I'll explain a bit what we mean by double fertilization events, okay? I'll show you what double fertilization events mean. Double fertilization events. Sorry, double fertilization, not events. Double fertilization. What do we mean by double fertilization? Double fertilization simply means, when one male gamete comes in contact with egg, okay? When one male gamete comes in contact with egg cell, and this is known as fertilization, right? Simply. Or we can say syngamy. And this results in what? Zygote formation, right? Zygote will be formed. We have not only one male gamete in pollen grain, we have two male gametes. So second male gamete will come in contact with polar nuclei, two nuclei, right? which are located in central cell, and they will form triploid cell, right? Triploid nucleus, and that is known as triploid fertilization, right? Three times nucleus will be formed. Fertilization or syngamy and triploid, three times what we call it as, sorry, the fertilization which will took place between two male gametes and polar nuclei, that is the both fertilization and that kind of syngamy is known as what? Double fertilization, because fertilization took place twice, right? Fertilization took place twice. That's why we call it as double fertilization, right? Let me clear it. Okay, that was double fertilization. What will happen now after double fertilization? After double fertilization, our zygote will get convert into embryo, right? Zygote was formed at the time of fertilization. When this zygote will get convert into embryo, it's known as post fertilization events. Similarly, when our ovary will get convert into fruit, that is post fertilization event, because this is the event which took place after fertilization. Similarly, when our ovule gets convert into seed, that is post fertilization event, right? And when our endosperm gets converted into triploid endosperm tissue, okay, that is known as what? Post fertilization events. All these events which took place in a flower or in a plant after fertilization are known as post fertilization events. And please remember, this endosperm development will precede, sorry, this endosperm this endosperm development will precede embryo development. Okay, this endosperm development will precede embryo development. Why? Because endosperm has to provide nutrition to the young embryo. Okay, it has to provide nutrition. Since it has to provide nutrition to the young embryo, it has to develop before embryo, right? So that when once embryo will start developing, it can easily provide nutrition to the growing embryo. I hope this concept is clear. Okay, now we will see, you know uh, how endosperm development will take place. We will see endosperm development. See what will happen. Say for example, this is our endosperm. Okay, this is our endosperm. What will happen to this endosperm? It will undergo rapid mitotic divisions in order to get increase in number. It will undergo mitotic division, that is equational division. Repeatedly, it will go under division and it will form cells like this. But it won't form cells, okay? Initially, it won't form cells. It will form what? It will form nucleus, okay? And these nucleus will be, in a single cell. Like cell wall formation will not proceed, will not be able to form after mitotic division. It has to be, first of all it will do only karyokinesis, then it will perform cytokinesis. Why? Because it has to grow in number. It will form like nucleuses like this, okay, inside the cell. And these nucleuses are known as primary endosperm nucleus, pen. Okay, these are known as primary endospermic nucleus. And primary endosperm nucleus will undergo successive nuclear division, which was in this nucleus is known as primary endosperm nucleus, and it will undergo so many divisions, successive divisions, and it will form free nuclei. Okay, and this stage is known as free nuclear endospermic division. This is known as This is known as free nuclear endosperm. Okay, I hope this concept of free nuclear endosperm is clear. And what will happen since so many nucleuses are formed? Subsequently, cell wall formation will take place. Okay, cell wall formation will took place, and these. If we will talk about your question might be, ma'am, in which we will found free nuclear endosperm and in which we will found cellular endosperm. Okay, you might have had sometimes in your lives coconut water, right? Coconut milk, which we call it as coconut milk as well. Okay, that coconut water is nothing but free nuclear endosperm. Okay, example of free nuclear endosperm, I'll write it down. Example of free nuclear endosperm. See what's that? That is coconut water. Okay, coconut water is the example of free nuclei, which has thousands of nuclei inside it, and the water we consume is nothing but nucleuses. Okay, and the white kernel you will see, in coconut, that is cellular endosperm. White kernel of coconut which we eat. Okay, that is white kernel of coconut is nothing but endosperm, cellular endosperm. Okay, cellular endosperm. I hope this concept is clear. Endosperm may be, what is the function of endosperm now? Function of endosperm is just to provide nourishment to the growing embryo. Now this endosperm embryo may completely consume developing, embryo might consume, whole endosperm, and that we have some examples in which we find no endosperm. We have no endosperm in that has been completely consumed by developing embryo. And the example is pea and coconut. Example is pea and coconut, in which endosperm is fully consumed, completely consumed. Okay, and in some we can say that in some, in some seeds endosperm persists. Endosperm inside the seed persists, and the example is castor and coconut. And the example is, sorry, it's pea, groundnut, and peanuts, in which embryo endosperm is free, completely consumed. But we have some examples, as I already mentioned, coconut. Okay, we have coconut in which what, endosperm persists. Okay, that means that is not fully consumed by endosperm, sorry, embryo. Okay, we have example of that is castor and coconut. Now, talking about embryo, as I already mentioned, zygote formation will took place at micropylar region. Okay, micropylar region where small pore is situated. Okay, and since at micropylar region zygote will be found, so can't we say that embryo development will also took place at, can't we say embryo development also took place at that micropylar end? Embryo development also took place at micropylar end, right? I hope this concept is clear. And you will see that most of the zygotes develop after the formation of endosperm. As I already mentioned, endosperm development will precedes embryo development, because endosperm has to provide nourishment to the developing seeds, to the developing embryo. Okay, but if we will talk about seeds, they differ. They are, different in monocotyledons and dicotyledons. Seeds, since monocotyledons have only single cotyledon and dicotyledons have two cotyledons, but their, embryogeny, or we can say stage of embryogeny, earlier embryogenic development is almost similar. Okay, how that is similar? Because embryo development took place like this. First, embryo will form proembryo. Okay, then embryo will form Then embryo will form globular embryo, globular embryo. Then embryo will form heart-shaped embryo. You can check the diagrams on NCERT. And finally, what will embryo form? Embryo will form mature embryo. I hope this concept is clear. Please remember, first of all, embryo will form proembryo, then globular embryo, then heart-shaped embryo, and then finally mature embryo. Okay. I hope this concept is clear. Since monocotyledons and dicotyledons are different, because monocotyledons contain only single cotyledon and dicotyledons contain two cotyledons. But their embryogeny, their development of embryogeny, which we call as embryogenic states, embryogeny, they are almost similar, because they will first of all give rise to a proembryo, then globular embryo, then heart-shaped embryo, and then finally mature embryo. Okay. I hope this concept of this is clear. Now, as we know, dicotyledons contain how many cotyledons? As the name says itself, dicotyledons contain two cotyledons, right? Dicotyledons contain two cotyledons, and it has embryonal axes. Since, say for example, this is one cotyledon, this is another cotyledon. Uh, the line in between the two cotyledons is known as embryonal axis. Okay, embryonal axis. On its lower side, embryonal axis has radicle. Okay, radicle, and on upper side it has plumule. Sorry, I'll write it down. On its lower side it has radicle. Okay, it has radicle, or we can say root tip, which is further covered. This is radicle, and it's covered by a cap-like structure. That cap-like structure is known as root cap. Okay, radical or root tip is inside root cap, and here we have plumule. Okay, this is plumule, or we can say shoot tip. Plumule or shoot tip, this is radical or root tip, which is covered inside root cap, and this is upper portion is known as plumule, or another name of it is shoot tip, which will give rise to shoot. Radical will give rise to root. Okay, I hope this concept is clear, how seed how it will be formed. And dicotyledon, since dicotyledon will have only one, sorry, monocotyledon will have single cotyledon, right? Monocotyledon, monocots have single cotyledon. And example of monocot is grass. Okay, and it has single cotyledon, so that single cotyledon is known as scutellum. This is known as scutellum. Okay, I hope this concept of post fertilization events is clear. I'll see you in such any other session. Till then, goodbye, take care, thank you so much.